Okay, let's start. So ayan, last week we already started on the idea of DNA, and we said that the primary function of the DNA or nucleic acids in general is for the storage of the genetic information. Okay? Sabi niya dito, it stores genetic information. In this genetic information is very important because it provides the instructions to the cells on what to do in order to maintain the different activities and homeostasis in the body. At the same time, it contains instructions on how to create certain proteins, certain amino acids, and certain kinds of cells, lalo na yung may mga specialized functions na cells. The instruction on how to make them is actually contained in our DNA. And of course, these traits or these instructions are being transmitted from one generation to another. That's why DNA is often related or connected dun sa idea of heredity. So, for example, yung certain traits such as the color of the eyes, height, color of the hair, everything, the texture is stored in our DNA. Pero yun yung kanilang function. Storage lang sila. But the actual making of these proteins and of these cells, syempre hindi na siya function ng DNA. Okay? And DNA is actually found most commonly in the nucleus of cells. Pero the nucleus is not the only cell organelle that actually contains its own DNA. There are two other organelles that was discovered to have their own kind of DNA. The first one is the mitochondria. So may sariling DNA ang mitochondria natin. And it's the reason why most of the traits that an offspring inherits comes from the mother. Kasi pag tinignan natin yung egg cell at saka yung sperm cell, in terms of size, mas malaki talaga yung egg cell. So basically, ang na-i-inherit lang ng offspring coming from the father is the DNA in itself, yung nandoon sa loob ng nucleus. Pero pagdating sa mother, ang na-i-inherit niya coming from the egg cell is not just the DNA but all the other organelles. So ibig sabihin, pag i-count natin yung DNA na nakukuha natin coming from our mother, we are inheriting not just the DNA found in the nucleus, but we are also inheriting yung DNA that is found in the mitochondria. That's why most traits na na-observe sa isang anak, most of the time, they come from the mother. Kaya nga sabi nila, it's very important that you choose, you are very specific or you're medyo taasan mo yung standard sa pamimili ng magiging future mother of your children. Kasi lahat halos ng mamanahin ng anak mo will be coming from them. Okay? For plants naman, the chloroplast also has its own DNA. And because of these organelles having their own DNA, scientists came up with the theory of what we call as endosymbiosis. So sabi nila, the original cell is actually ito daw yung ancestral eukaryotic cell. Ang meron lang siya is itong nucleus that has its own DNA. And then somehow, it was able to engulf or ipinasok niya sa loob ng eukaryotic cell itong mga smaller cells that originally were called as aerobic bacterium. Ibig sabihin, yung origin daw ng mitochondria dati, because they were smaller, they were theorized to be bacteria that are able to do cellular respiration. At nung nakita nitong ancestral eukaryotic cell na magiging advantageous sa kanya kung meron siyang sariling way of using oxygen to produce 38 ATPs. Ang ginawa niya, ipinasok niya sa loob ng cell niya itong aerobic bacterium na to. And symbiosis, this is a kind of ecological relationship. Wait lang daw. Okay, going back, if you will remember sa ecological relationships, meron tayo yung predation, parasitism, mutualism. Okay, so itong endosymbiosis is actually a kind of mutualism wherein both the ancestral eukaryotic cell benefits from the aerobic bacterium, and the aerobic bacterium also benefits from the ancestral eukaryotic cell. Ibig sabihin, both of them are receiving benefits from each other. That's why it worked for them. So for the aerobic bacterium, so syempre, habitat, safe siya sa loob ng ancestral eukaryotic cell. At the same time, mas readily available na yung food or source of energy doon sa ancestral eukaryotic cell. Pagdating naman sa ancestral eukaryotic cell, ano ang nabibenefit niya doon sa bacterium? Siyempre, it's the conversion of the food na meron siya into a huge amount of ATP through using the availability of oxygen. So because they work well together, eventually, the eukaryotic cell, hindi na siya nagre-reproduce ng walang mitochondria. So eventually, nung nag-evolve siya, they became the same part of a singular cell. So ayan na yung, anong tawag dito? Ito na yung available na animal cell na nakikita natin ngayon, na merong nucleus at the same time, meron din siyang mitochondria. Pero nung sabi nila, based on their theory, yung pinakaunang eukaryotic cell, wala talaga siyang, kumbaga hindi automatic sa kanya na meron siyang mitochondria. Okay? And then this eukaryotic cell eventually evolved as well. So hindi na enough para sa kanya na nakakapag-convert siya ng maraming amount of ATPs in the presence of oxygen. Ngayon naman, nakakita siya ng isa uling bacterium. This one is a photosynthetic bacterium. It has its own DNA and it has its own ability to produce food using the energy coming from the sun. So, kinuha ulit niya, pumasok ulit yun doon sa eukaryotic cell. So, eventually, yun na yung naging plant cells. So, itong theory of endosymbiosis na to, it came from the discovery of the scientists na merong mga DNA itong mga certain organelles na to. Meaning, if they have their own DNA, they are able to survive on their own. They are able to exist on their own kasi kaya nilang mag-replicate, kaya nilang mag-pass on ng traits from parent to offspring from one generation to another. So because they have this ability, yun nga yung naging theory nila na even before, nag-i-exist na itong chloroplast in the form of a bacteria, the same itong mitochondria, nag-i-exist na sila noon in the form of a bacteria, mas maliit sila doon sa eukaryotic cell. And then eventually, because they are able to work together through the idea of mutualism and endosymbiosis, they were able to form a single cell that can produce all of the needs of the organism. It can create its own food, and then pag nagawa na niya yung food na yon, it can convert it into a huge amount of ATP. Okay, so again, what is this slide trying to tell us? It's trying to tell us na hindi lang sa nucleus nakikita ang DNA. There are other organelles that have their own nucleic acid, that have their own DNA, which allows it to pass the traits from one generation to another. Okay, so let's continue. This one is a review basically of what we learned last week. So the structure of the nucleic acid DNA, sasabi natin, DNA stands for deoxyribonucleic acid. So coming from the word, meron siyang deoxyribose, which is a sugar. And then this sugar is attached to a phosphate group. And this sugar and phosphate, it acts as the backbone of the DNA. And then in the middle, there are nitrogenous bases. The nitrogenous bases are divided into two. we have the pyrimidines, and we have the purines. Before I continue with the next slide, can somebody tell me what's the difference between pyrimidines and purines? Yes, Yunila.
Yung purines po, double ring po siya. Yung isa naman po, single ring po.
Okay, thank you very much, Yunila. So sabi niya, purines, these are nucleotides that are double-ringed, whereas your pyrimidines are the nucleotides that are single rings. Can anybody tell me what are the two purines? Okay, may nag-chat. Adenine and guanine is your purines. How about the pyrimidines? Of course, the remaining two, it's thymine and cytosine. Okay, so ito yung purines natin, adenine and guanine. So based on the description of Yunila, double rings daw sila. So if you will notice here, adenine has this hexagon ring, and then meron siyang pentose ring dito, dalawa, double ring. The same with guanine, meron siyang hexagon ring, and then meron din siyang pentose ring. On the other hand, you also have your pyrimidines. These are the single ring na mga nucleotides or nitrogenous bases. We have thymine and adenine. So kung mapapansin nyo, iisa lang yung kanyang ring. It is just one hexagon ring. Okay? So if we are going to combine the DNA backbone, which is composed of the deoxyribose and the phosphate, naka-attach dyan yung nitrogenous bases. Okay? So the nucleotides, in terms of the connecting bond between the deoxyribose and the phosphate, then it is It is composed of a covalent bond, specifically yung phosphodiester bond. Okay? Pagdating naman sa nitrogenous bases between a pyrimidine and purine, ang kanilang bond is what we call as a hydrogen bond. So sabi natin, very important na hydrogen bonds yung makikita natin between the nitrogenous bases, because hydrogen bonds are the weakest form of bonds in chemistry. The strongest is always covalent bonds. And later on, we will learn kung bakit importante na mahina lang yung bonds that are connecting these nitrogenous bases. Kung bakit advantageous na hydrogen bonds ang available doon sa nitrogenous bases. So this is our DNA. It is composed of two strands. This is the backbone. The backbone is composed of the sugar phosphate, the deoxyribose, and the phosphate group. And then in between them are the hydrogen bonds connecting the two nitrogenous bases. So ang ating connection is always one pyrimidine that is connected to a purine. And between adenine and thymine, we have two hydrogen bonds. And between guanine and cytosine and vice versa, we have three hydrogen bonds. So kung makikita ninyo dito in this picture, itong dotted lines na to, all of these are hydrogen bonds. And pag tatlo yung line niya, yung dotted line, then it means there are three hydrogen bonds connecting cytosine and guanine and cytosine. Whereas in adenine and thymine, there are only two hydrogen bonds connecting them. Okay, let's continue. So, ayan. The same, ito yung phosphate group, itong may red na circles. The deoxyribose is composed of the black circles. Ito yung carbon. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And then we also have the nitrogenous bases here connected with the hydrogen bond to another nitrogenous base that is connected to a sugar and a phosphate. Okay? So paano nga nila nalaman yung pairing? Kasi if you will notice, when it comes to the DNA, meron silang tinatawag na pairing. Ang magka-pair lagi is adenine and thymine or thymine and adenine. Tapos, the other two, yung kanyang cytosine and guanine or guanine and cytosine is always partnered up. So the scientist who discovered this complementary base pairing idea is who we call as Erwin Chargaff. So what he did is he studied different organisms. He isolated DNA from different organisms, specifically in terms of Streptococcus, tapos yeast, herring, and then human. So medyo from smaller organisms to bigger organisms yung representatives niya in his experiment. Streptococcus is a bacteria. Yeast is a unicellular organism na eukaryotic na siya. So ito yung ginagamit na pampaalsa. Most of you in your fermentation use yeast. So that is a unicellular eukaryotic organism. Herring is a form of bird. And then of course, we have humans. Okay? And ang ginawa niya, dun sa na-isolate niya ng mga DNA, pinaghiwa-hiwalay niya yung percentage ng adenine, thymine, guanine, and cytosine. So pwede na siyang gawin ngayon. Even during my time, as early as 2006, 2007, may mga experiments na kami or may mga thesis experiments na sa amin na regarding DNA. Tas parang nag-e-encode sila ng DNA sa isang organism. Kasi DNA encoding is actually used for the classification of organisms. So itong si Chargaff, ang napansin niya, magkakalapit daw yung amount ng adenine and thymine. At magkakalapit din yung amount ng cytosine and guanine. So for example nito, adenine is in Streptococcus is around 29.8% of the DNA isolated. Tapos itong thymine is around 31.6. So magkalapit yung kanilang percentage. Whereas itong guanine nasa 20.5, ang cytosine ay nasa 18. So magkalapit din yung kanilang percentage. So ang naging idea niya is since sila yung magkalapit ng amount, ibig sabihin, sabi niya, most likely sila yung laging magka-partner. Kaya kapag merong adenine dito, automatic sa kabila, thymine. Pag may thymine dito, automatic sa kabila, adenine. Kaya sila yung magkakalapit ng values or percentage in a DNA. Hindi kasi pwedeng ang magka-partner ay adenine at saka guanine. 30 ang adenine, yung guanine ay 20. So ibig sabihin, pag sila yung magka-partner, may mga adenine na wala siyang ka-partner. Walang guanine kasi hindi enough yung guanine na nandoon sa DNA para masabayan niya yung dami ng adenine na nandoon sa DNA. So yun yung line of thinking ni Chargaff, which allowed him to form the conclusion na yung magkakalapit ang amount, yun ang magkaka-partner. So he gave the idea of complementary base pairing na adenine and thymine ang laging magka-partner at cytosine and guanine ang laging magka-partner. So he did that to Streptococcus and he checked if yung hypothesis niyang yon will also be observed in higher forms of organisms. So from Streptococcus, chinek niya yung yeast and he also found the same results, magkakalapit, 31 and 33 yung adenine and thymine, in 19 and 17 yung cytosine, so magkadikit pa rin. So hindi pa rin pwede na adenine at saka guanine ang magka-partner. From 31 to 19, diba? So ang daming mga blank spots in the form of adenine kung sila yung magka-partner. He further tested his hypothesis with herrings and humans, and he also found similar observations. So he therefore concluded na ang magka-partner talaga is always adenine and thymine at saka cytosine and guanine. And even up till now, ginagamit itong concept ni Erwin Chargaff in classifying organisms. So may mga organisms, lalo sa mga halaman, na magkakamuka yung itsura nila. Pero if we are going to analyze them genetically, molecularly, ang ginagamit ngayon ng scientific community is the percentage of cytosine and guanine. So pag magkakalapit yung kanilang, anong tawag dito? Yung kanilang percentage, ibig sabihin, they belong to the same family. Pero pagka magkalayo yung kanilang percentage, ibig sabihin, they don't belong in the same family. So ngayon, the molecular basis for classifying organisms is based on the amount of cytosine and guanine. Okay? So ito na yung ating DNA. So magka-partner is adenine and thymine. Pag thymine dito, automatic adenine sa kabila. The same rule applies to guanine and cytosine. Okay? And we also discussed this last week, the idea of antiparallel strands. So when we talk about this idea of 5' and 3', what we are talking about is the direction of the strand. So nakadepende siya sa kung saang carbon naka-attach yung ating phosphate group. So on this strand of the DNA, itong phosphate group ay naka-attach doon sa 5 or sa 5th carbon. Kaya dito ang tawag sa kanya ay 5'. So 5', 3', 5', 3', 5', 3'. So yung dulo niya is always 3'. So ang direction of movement niya is from 5' going to the 3', which is the exact opposite on the other strand. Kasi baliktad siya, di ba? Hindi sila iisang direction ang pinupuntahan. Anti-parallel sila. Ibig sabihin, they're going against each other's direction. So kung 5' to 3' itong isang strand, automatic, yung kabilang strand niya is the opposite. It will start with 3', so doon naka-attach yung kanyang phosphate group, nasa 3', and then ang nasa baba niya ay yung 5'. Okay? And this is important later on. We will learn na the process of replication follows a certain direction of movement. Okay, so now let's focus on the idea of DNA replication. So sabi niya dito, kailan ba nangyayari ang DNA replication? Nangyayari ang DNA replication during the interphase stage of the cell. Specifically, di ba yung interphase, merong tatlo. Merong G1, may S phase, at saka G2. DNA replication specifically happens in the S phase. Kasi di ba in the S phase, ang ginagawa ng cell is that it has to double its DNA. Tatandaan natin, during cell division, we are starting from one parent cell and it has to produce 2 daughter cells which will have the same exact DNA. So mitosis, di ba? Walang pinagbago yung DNA ng parent at saka ng daughter cells. So kung iisa lang ang parent cell, paano niya ma-ensure na yung dalawang daughter cells niya will have their own copies of the DNA? So ang gagawin niya, kailangan muna niyang doblehin yung DNA na meron dito sa parent cell. So during the S phase, the DNA replication happens. So from 2 copies of the DNA, magiging 4 copies siya para at least each daughter cell will have their own set of genes. Okay? So during the S phase, dodoblihin niya yung kanyang DNA sa loob ng nucleus para pag nag-divide na siya, parehong daughter cells will have their own copies of the genes. Okay? So DNA replication happens during the S phase. It happens in all kinds of cells, specifically yung ating somatic cells, which undergoes mitotic cell division. Hindi lang nangyayari, ang DNA replication nangyayari rin sa sex cells, pero not all the time. For example, sperm cell, pag dumadami sila, that is still mitosis. So DNA replication pa rin ang ginagawa nila. Okay? And specifically, DNA replication happens in the nucleus. So important to kasi later on we will find out that transcription and translation actually does not happen inside the nucleus. So only DNA replication happens inside of the nucleus. Okay, and there are 3 steps to what we call as this DNA replication. Okay, so the first one is of course the unzipping of the double strands. So hindi pwedeng kopyahin yung isang strand if hindi maa-unzip itong double strand na to. At dito pumapasok yung importance na hydrogen bonds ang meron in between your nitrogenous bases. Kasi kung covalent bond yan, ibig sabihin mahihirapan yung cell na i-unzip itong double strand na to. And the unzipping of the double strand, kaya siya tinawag na unzipping kasi para siyang zipper na, di ba, pagka yung may gitna, nandun na siya, saka siya naghihiwalay. So, ganun yung nangyayari dito sa double strands ng ating DNA. And the unzipping is made possible by an enzyme that we call as the DNA helicase. Okay? So, kaya ang tawag sa kanya ay helicase because it is an enzyme that breaks the hydrogen bonds between the 2 strands of the DNA. Okay, so kung nakikita niyo dyan sa picture, yung kulay green is actually the hydrogen bonds, and this is broken down by what we call as the DNA helicase. Okay, so kapag na-unzip na yung double strand of the DNA, papasok na itong si DNA polymerase. So, DNA polymerase is another enzyme, pero ang function niya is to attach free nucleotides into the complement and form complementary bases with the original strand. Balik tayo. Saan ba yan? Sige, ito na lang. Okay? So, kung mapapansin niyo, ito yung mga free nucleotides. Ibig sabihin, they are just floating freely inside the nucleus. And then, nung nag-unzip na siya, ang na-expose na nitrogenous bases is, for example, kung adenine siya, ang ikakabit ng DNA polymerase ay thymine. On the other side, ang ikakabit naman nitong DNA polymerase ay adenine. Okay? And then, pag naikabit na yon, of course, it's also the responsibility of the DNA polymerase to form hydrogen bonds between these nitrogenous bases. Okay? So, and then of course, pagka nakakabit na yung nitrogenous bases dyan at okay na yung hydrogen bonds between them, syempre kailangan na rin mag-join nitong kanilang mga, anong tawag dito? Yung kanilang mga backbone. Tatandaan natin, doon sa step number 2, ang nadagdag lang ay yung nitrogenous bases. So, ibig sabihin, yung isang strand, wala pa siyang backbone, wala pa siyang sugar, at saka wala pa siyang phosphate. So, yun naman yung trabaho nitong tinatawag nating ligase. The ligase will now attach the sugar and the phosphate doon sa mga nitrogenous bases na naidagdag. And of course, it has to be connected with a covalent bond. So, itong covalent bond na to, again, specifically, it is a phosphodiester bond. The bond between the sugar and the phosphate, which is the backbone of the DNA. So, pag itong ligase na ikabit na yung sugar and yung phosphate, ibig sabihin, kompleto na. Instead of 2 strands, meron ka ng 4 strands. At yung 4 strands na yon is composed of the old strand and the new strand. So itong purple here is the old strand. Okay? And then dinagdagan siya ng nitrogenous bases, dinagdagan siya ng sugar and phosphate. Ito yung kulay red. This is what we call as the new strand. On the other side, ganun din. Yung isang strand niya is an old one na dinagdagan ng hydrogen, ng nitrogenous bases, dinagdagan ng sugar and phosphate. So now, it has another strand of DNA. Ito yung red. So dun sa 4 na strand na yon, 2 ang old strand at dalawa ang new strand. So 50-50%. 50% is old, 50% is new. And this is the idea of what we call as semi-conservative replication. Ibig sabihin, kalahati na co-conserve niya yung original na strand. So in this new strand, or in this daughter strand, yung kalahati niyan is an old strand of the DNA at yung kalahati niya ay still a new kind of DNA. So 50-50%, semi-conservative replication. So yung kalahati pinaggayahan, yung kalahati yun yung ginaya. Okay? So in this picture, ayan, the first thing that happens is that this helicase will unzip the DNA strand. At pag na-unzip niya na itong DNA strand, the DNA polymerase here will start attaching the free nucleotides. So for example, itong kulay blue, ang kapartner niya ay yung green. So yung green, ang kapartner niya yung blue. Yung red, kapartner niya orange. Ang idadagdag dito, green, green, red, orange, green. Okay? So pag na-add niya na itong mga nitrogenous bases, the ligase will now attach this, ang tawag dito, itong backbone niya. So pag na-create na yung backbone, now you have the complete daughter strand. Na itong kulay light blue is the original strand, the old strand. Yung darker blue is the new strand. Okay? And is it in the next slide? Wait lang. May nag-chat. Yes po, they work simultaneously. Wait lang nasan yung arrow ko? What happens in the DNA kasi, oh my God. Jordan, wait lang. Mag-i-end ang aking